Okay, I always like to start by asking this question. When is the last time you hiked through a chestnut forest? When you hiked through a chestnut forest, when was the last time you did that? Most of you say, why are you asking that question? There is no chestnut forest, right? Well, that's a shame because at one time, the American chestnut tree represented 25% of the standing timber in the eastern United States, and that is no more. So what I'm going to be presenting to you today is our research at the SUNY College of Environmental Science and Forestry, and it's entitled New Technologies Solving Old Problems. The chestnut was lost over 100 years ago, or started being lost over 100 years ago, and we now have an opportunity to actually start bringing it back. Okay, so why chestnut? Why did chestnut work? Well, chestnut really considered one of the most important keystone species in the eastern forest. And why do I say that? Well, mainly because of the nut crop it produces. It produces a nut crop that's very consistent from year to year. So it produces a nice mass for wildlife. Since we've lost the chestnut, the trees that have replaced it are mainly the oaks, which are not consistent from year to year. The reason why there's a difference, oaks flower earlier, and so they're susceptible to late frost. American chestnut flowers later, and therefore is resistant to late frost. You say, okay, nut crop. So why is that important? Well, you know, a lot of wildlife like it, and not only squirrels, but raccoons, bears, deer, wild turkey, blue jays, all different kinds of wildlife like to utilize American chestnut. Now, that's just the nuts. But there's a lot of other things about chestnut that help support the forest. For example, the leaves. There's a collaborator, Vern Sweeney, who's looking at leaf litter in streams. And what he's finding is that American chestnut leaves are preferred over the oaks that have replaced them for the insect larvae that live in those streams. They prefer it, they grow bigger on it. So why is that important? Well, of course, that's important because the things that eat the insects, the fish. So the American chestnut actually has an indirect impact even on fish in the streams and the mountains. Now, American chestnut also has an economic value. It has an agricultural value. You probably know this time of year, think about roasting some chestnuts. But chestnut actually could be ground into a flour and used for baking. Thoreau in Walden actually said that chestnut is a suitable substitute for bread. And you can actually make bread. You can take chestnut flour and actually brew it into a beer. And the nice thing about that is actually a gluten-free beer. For those of you who are like me, you can't eat gluten. Chestnut has a timber value, or a wood product value. Chestnut wood is very straight-grained and it's very rock resistant. It's kind of interesting. You know, telephone poles are made out of wood, or at least most of them are. The longest used telephone pole was made out of chestnut, over 80 years. And it was not treated. It uses rock resistance. So you could use this for purposes where you wouldn't have to treat the wood. I always tell people, if chestnut was abundant today, everybody would be making their decks out of chestnut because it was rock resistance. Again, very straight-grained wood, very easy to work. My grandfather, who was an antique dealer, said that, you know, when you look at antiques from the 1800s, you don't find too many with chestnut on the outside. Where you find the chestnut is on the inside. Okay, it's often used for like the inside of drawers, all the things, because it was very easy to work. Not quite as hard as a walnut or a black cherry, but great for interior parts. Now, American chestnut is really part of our heritage here in the United States. And you can't go to a town without finding a chestnut street. I should have looked. I don't know if Ithaca has a Chestnut Street, but it probably does. I particularly like this intersection for obvious reasons. This is actually out in San Diego, you know, all the way across the country. And so people have this knowledge and background of the American chestnut tree. This time of year, you'll be hearing a lot of Christmas carols. One of them, you might hear chestnuts roasting on the open fire. That's actually talking about the American chestnut. Now, I don't know how many people get up to Syracuse Way, but up above Onondaga Lake, we have a park called Long Branch. lot of people don't realize it, but Long Branch Park was actually named after the chestnut trees that were there. You know, so chestnuts even ingrained underneath the obvious, so part of our culture. And as I said, people all across the country know about chestnut. We are doing a project right now to try to raise some funds to reduce our trees. And one thing that's interesting about that is that we are looking for donations. I'm not asking anything for you right now, but I just want to show you this map. In 2 weeks' time, we've had donations from all across the United States, mostly in the natural range of American chestnut, but all the way out west of California, even in Alaska, and even out to Hawaii. We had donations coming in from New Zealand, from the Netherlands, from Brazil. So a lot of people realize that chestnut is important. Now, the American chestnut sometimes is called the redwood of the East, even though it never gets as big as a redwood tree. But it is one of the largest trees in the eastern forest, or can be one of the largest trees in the eastern forest. This is the classic picture of the lumberjacks standing around some American chestnut trees. You can kind of visualize how large they can get. 80 feet, typical, even over 100 feet occasionally. 5 to 8 feet diameter, typical, sometimes reaching as much as 10 feet in diameter. Another way to kind of look at this, I like this. This is not a photograph, obviously. Well, it is a photograph. It's a photograph of a painting called the Chestnut Harvest. And I just like to show this because these are people down here collecting chestnuts. And to kind of give you a perspective of how big these trees are, there's a guy up there knocking the chestnuts down to the people below. Okay? So these trees could get very, very large. Now, are there any large chestnut trees left? Well, there are a few, and they usually are the trees that people have planted outside its natural range, and therefore is protected from chestnut blight, which I'm going to talk about in a minute. This one here is actually in Michigan, and it kind of gives you an idea of how large these trees can get. You can see it's at least twice as high as this two-story building, and it has about a 5-foot diameter base, and very nice tree. Unfortunately, this tree is not around anymore. A few years after the person took this picture, he went back and looked at it again, and this is what happened. Okay? The people there actually cut the tree down to use it for firewood. You know, they didn't really realize what they had. They just had this big old tree out there. It was probably not producing nuts because it was a lone tree, and chestnut has to have 2 trees next to each other to cross-pollinate and produce viable nuts. So they got rid of it, unfortunately. So why don't we have chestnuts everywhere? Well, we don't have them because of an exotic pathogen that was introduced in this country called Nectar parasitica. It's a fungus. And this fungus is a necrotroph, and what it will do, it's a very happy pathogen, as you can see. That was actually a picture taken by my technician, Andy Newhouse, and that's actually what it looked like on the plate. So this fungus, what it will do is it will enter a wound in the tree. It could be just a branch scar, such as that. It will colonize that wound, and then its physiology changes. And it changes in a way that it starts forming mycelial fans, which are kind of like mechanical wedges that help it pry into the tissues of the tree. But more importantly, it starts producing a number of acids. The main one is oxalic acid, or oxalate. And that acid will actually kill the tissue ahead of the spreading mycelium and allow it to utilize the dead tissue. But when it's doing that, it's forming what's called a canker. And this sunken orangey area is the canker, which will eventually girdle the tree, cutting off all circulation up and down, killing everything above it. Once you get a canker on the base of the tree, it gets killed down to the ground. Okay. Now, when the blight first came through, it really kind of caused a panic. And you'd see headlines like this. You know, all chestnut trees here are doomed. Chestnut trees face destruction. These are some of the actual headlines from back in around 1904 and earlier, or a little bit later. So people really were wondering, what can they do? How many people here are actually from New York State? Okay, no, about maybe a third of you. The interesting thing about New York State is we have the distinction of this is where the blight actually started. We are ground zero for the blight. And we know that because the blight came from Asia on imported Japanese chestnut trees. And we know that through historical records that in 1876, a nurseryman, S.B. Parsons, started importing the first Japanese chestnuts. Now, we don't know if he had the blight on that first batch of trees, or actually they were nuts, the first ones he brought over. So we don't know if that was on there. But sometime between 1876 and 1904, when the blight was first described in the Bronx Zoological Park, the blight had come over. Right? And within 50 years, by about 1950-something, the blight had moved from its original introduction up here and moved through its entire range, killing somewhere between 3 to 5 billion large American chestnut trees. Now, to kind of get you a picture in your mind, because it's really hard to visualize just how prominent these trees were. This is a very rare photograph of a chestnut forest. This is at the Shenandoah National Park down in Virginia. And what you're looking at here is a chestnut forest. This is in the spring, so all these kind of like highlights here, those are catkins, or the male flowers of the chestnut. And it's all in bloom, and you can see it's the predominant tree in that forest. And these are very large trees. This was taken in 1912, just before the blight reached that far south. But what was happening in New York in 1912? Well, this is the old photograph along the Long Island Railroad. And what people were seeing as they were traveling down that track is they look out the window and see mile after mile of dead trees. And these are the largest trees in that forest. Okay? This is not taken in the wintertime. This is a summer photograph, so you have some trees are fine with leaves, but these are all dead American chestnut trees. So it was very, very severe. Do we have anything like that today? Well, probably nothing that comes close to the chestnut blight. But we do have some pretty severe pest problems and pathogen problems out there. I was going to throw this one out here. This is woolly adelgid on hemlock. And this is a picture from North Carolina along the Blue Ridge Parkway. I took this, and you can see all these gray trees in here. These are dead hemlocks. So picture that and at least double it, and that's what the chestnut blight would have been like.
Hemlock or Carolina?
I didn't go down and actually identify them. I'm not sure which they are. Okay, so does that mean American chestnut is extinct? Nope. And the reason why it's not is because of two reasons. One is that the fungus itself cannot compete with the soil microorganisms, and also because chestnut has the ability to sprout from the root color down here. So those combination of things actually has allowed the chestnut to survive, and that's a good thing because in a restoration program, we need to have a lot of the population to recoup a lot of the genetic diversity that was out there. And there are literally millions of these stump sprouts still surviving today. So what's basically happened, oh, and I just want to mention too, these stump sprouts are very vigorous because they have a nice root system to them. And if you were to cut off or coppice a chestnut tree, you could actually get about 7 feet growth in a single season. This is what we did with this particular tree. You cut it, it grows 7 feet. So these things will grow very robustly for a few years until they finally get the blight and get killed back down to the ground again. So the chestnut is kind of caught in this type of cycle. Anybody remember Sisyphus from Greek mythology? This is the guy who was doomed to roll a rock up a hill only to have it fall back down and have to repeat the cycle. That's what the chestnut is doing right now. It's where it's growing for a time, it will get the blight again and get killed back down to the ground. Okay. Now, the American chestnut was so important that there has been a lot of different attempts to try to rescue that tree. But up to this point, really nothing has worked, even after 100 years of research. Now, you might hear people saying that, oh, we have trees that we can plant out to replace the American chestnut. For example, if you go on the web and you might see something like this, the Dunstan hybrid chestnut. Okay? It's claimed to be the most widely planted chestnut tree in America. Well, the Dunstan hybrid is a cross with American, but what they've done is they crossed American chestnut with 3 different lines of Chinese chestnut and then intercrossed all those. Okay? So it's definitely a hybrid. It's not an American chestnut, and I'll tell you why that's important in just a minute. Some people even gone further and not just crossed with American and with Chinese, but they've crossed every type of tree type chestnut out there. They've crossed American, Chinese, Japanese, European, and Saguenay chestnuts and mixed those together and interbred them and try to select traits that they liked. Okay? Now, this is fine. These trees are okay if you want to have a tree as an orchard tree or something you can plant in your yard, but these trees will not survive in the forest. They are not adapted for our eastern ecosystems, and they will not grow like the American chestnut. Okay? Now, there are a couple programs that are trying to regain the characteristics of the American chestnut, and they're having some success. One is the Backcross Breeding program at the American Chestnut Foundation. And this goes a little different than the typical hybrid breeding. What they're doing is they first take a hybrid, which is Chinese and American, and then they backcross it through 3 generations to recapture those American traits that will make it more suitable for our forests. At the same time, of course, they have to retain the resistance from the Chinese chestnut, which is quantitative, and it involves at least 3 genetic loci, possibly more. So it's a very complicated thing to do. Also because chestnut has a long, can't think of the term now, period between nut to flower. They are going through 3 generations as opposed to a typical maybe 6-generation backcross system. So you're going to have a lot more of the Chinese characteristics in there than you would normally. So, but the goal here is that you're going to have 1/16 Chinese, 15/16 American, and hopefully retaining the 3 or more blight resistance genes, which is quantitative. You also have to have them in a homozygous state to get full levels of resistance. Complicated, but they are making some success. They have some from the 3rd generation backcross trees that are actually quite resistant. What they haven't gotten to the point yet is where their offspring will also be resistant. What it is, they get offspring that basically span the range of resistant to susceptible. So, but they're on their way with this. Now, we're doing something different. We're doing genetic engineering, or transgenics. And I just want to compare the breeding to the transgenics. Both are viable, but transgenics have certain advantages, and I just want to show you some of those advantages. Now, we know that chestnut has about 45,000 genes in it. How do we know that? Well, we're in the age of genomics, right? Everything's being sequenced. Chinese chestnut has been sequenced. American chestnut is getting to be sequenced. So we kind of know what the number, anyway, of genes that are present. So you got to think about this backcross breeding first. If you're backcross breeding and you still have 1/16 Chinese genes, how many genes is that? It's quite a few genes still. Is that a problem? Maybe, maybe not. The question here is, in those 1/16 Chinese, are there traits that still make it unsuitable for our forests? Is it going to be too short? Is it going to be cold hardy enough, which Chinese chestnut's not as cold hardy? Are the leaves going to be preferred by the insects, like the American chestnut? We don't know, and a lot of different things. So what I like to do when I describe this to the general public, I like to relate this to a book, okay? You think of a book, and you think of the words in the book as genes. So if you have a book that's 1/16 Chinese, that means you have about 11 pages or 2,800 or so words in Chinese. Now, that might not matter. If those things are spread out, you might be able to read that book and still get the gist of it. But if some of those words is in a critical plot line, you might miss whodunit, right? So it really depends where those genes are. And so that's what you have to do, a lot of work to make sure that those genes aren't going to matter while you still retain the resistance genes. Now let's look at transgenics. Again, looking at this book example. So we're still going to look at the whole book, but now we're just going to pull out half a sentence here. And that sentence says, "It was very exciting at that season to roam the then boundless chestnut woods of Lincoln." I like this because this is from Thoreau, Walden, talking about chestnuts. So in transgenics, we're making very small changes, only adding 2 to 4 words to that. So if you use this book example, it would come out like this. In this whole book, it was very exciting at that season to roam the then boundless blight-resistant chestnut woods of Lincoln. Okay? So the advantage here, of course, is we're adding blight resistance, and we don't have to get rid of anything else because it's not there to start with. Okay? So it makes things a little bit simpler. All right? Now, that's just a thought experiment. Let's actually look what's happening in the trees. And we are starting to do some metabolomic studies. Metabolites are the small molecules in the cells that are indirectly related to the change you make, because they are made by the enzymes, they're encoded by genes. They can be like amino acids, fatty acids, phenolics, those type of things. Well, we did some studies. We picked 112 to represent kind of a broad range of metabolites. In this particular example, we're just going to look at the leaves. And if you were to compare American chestnut to Chinese chestnut, now we're comparing the same metabolites in both, just looking at different levels of expression. And sometimes the levels can be as much as 200-fold different. Okay? So in this, Chinese is related to American, so you have about 2/3 being about the same, about 1/3 being different. Okay? Now, if you start a backcross program, you actually start recovering more of the American type here. This is a Backcross 1 hybrid chestnut, and you can see you have much more similarity to American than the Chinese here. But let's look at the transgenics. Okay? Here's the transgenics comparing the American chestnut to the transgenic chestnut. Out of these 112 metabolites, only one has changed. Okay? Kind of confirming that, yes, we are making a very small change in this tree. So what is that change? You say, well, it's one change, but what if it's a really important change, right? Well, let's dig down a little bit. The change is actually in alpha-linoleic acid, which is a essential fatty acid. And if you look at the transgenic, it has slightly less than the non-transgenic on average, about a 1.6-fold difference. But if you compare that to the backcross tree, it's no different, and it's actually no different than Chinese. So is this really significant? Probably not. It's not biologically significant. So again, we're making a small change, and the changes aren't any different than what you're seeing with the breeding. Okay? Okay, so I'm going to take a minute and explain how do we get a gene into the tree. And I'm sure a lot of you here know about this already, but I'm going to go through it. We use Agrobacterium tumefaciens, which is a natural genetic engineer. It's been doing this for millions of years in plants. And basically what it does is it inserts a piece of DNA into a plant cell, and it kind of hijacks that plant cell so it starts making food that only it can utilize, right? So what scientists have done is they've tamed the Agrobacterium, and instead of it putting the gene it wants to put in, it will put the gene we want to put in, right? So, but here's the problem. It puts it into one cell at a time, right? So you can't use Agrobacterium and transform a whole plant. What you have to do is transform a cell, and you have to be able to regenerate a whole plant from a cell. And this, for chestnut, took up most of our 25 years of research, because it was not easy to do. So I'm going to describe that real quickly in a series of slides, how to make a tree from seed the hard way. Okay? Now, chestnut, we cannot regenerate from leaf slices. We cannot regenerate from meristems. We have to actually go back to embryos. And there's about a 2-week window when you can collect immature embryos, cut them open, dissect them, sterilize them, and you end up with isolating the embryos from inside that immature nut. There's actually quite a few embryos in there. Some of them will be fertilized, some will not. Usually you get a dominant one. You got one here that's dominant that will eventually end up being the embryo for the nut. But we plate those out on a special media to try to induce somatic embryogenesis. And somatic embryogenesis is basically where you grow embryos and they bud off new embryos. Right? And only about 1 in 1,000 of these actually will develop into a somatic embryo culture. Now, why is that important? Well, it's important because these embryo cultures, every one of these little embryos that bud off, all originate from a single cell. So if you transform that single cell, you can get the whole embryo. Right? And these things can be maintained for a number of years. We have several that have gone for almost a decade. And that's the material we use for the Agrobacterium transformation. When we first did this, a number of years ago, it took us 24 months from transformation to you actually had a potted plant. So you had to be very patient. We've, over the years, cut this down to 18 months, and now it's actually down to about 12 months. So we've gotten better at this transformation methods. Okay, now, at this point, I want to introduce this term that some of you know, but some of you might not, and that is event. Some of you might think an event is like a rock concert. We're not talking about that. We're talking about a transgenic event. And the reason why this is important is that this is not only important biologically, but but it's important for the regulators who's going to review our trees in the future. They will deregulate by the event. So let me describe what that is. An event, what we have here is a somatic embryo culture. This is being looked at under a microscope with a fluorescent microscope. And when this light green in the background is normal background fluorescence, the bright spots are caused by a marker gene we use called green fluorescent protein. And I'm just going to use this to illustrate what an event is. So if you were to transform this particular cell and grow up an embryo culture and make shoots and trees, everything that comes from that cell would be one event, okay? And then be clonal to that event. But if you grow up from this cell over here, that's going to be a different event, okay? So, but you can make hundreds of trees from that event, but it's going to be different than the first one, okay? Now, why is that important? Well, it's important because when you do agrobacterium transformation, the genes go in what I like to call in a semi-random fashion. And what I mean by that is it tends to go into the euchromatin, the unwound pieces of DNA, more than the heterochromatin. And so it's not completely random, but it's semi-random. And what that means is it can go into different places. So the way I like to envision it is, let's say you had the 12 chromosomes in Chestnut. If you had the gene insert in this chromosome here, that would be one event, and then everything produced from that is going to be in that spot. But that other event, maybe it went in over here, and everything you produce from that is going to be over there in that spot. Now, why is that important? It's important because of what's called position effect. Your neighbors influence your gene expression, right? And the way I like to people visualize that is to think about real estate. It's always location, location, location. So let's say you build a house here in Ithaca, and it's worth $150,000. If you were to build that same house in San Francisco, it might be worth $400,000, even though it's a clone of that house, made with exactly the same floor plan, same materials and everything, but just because of location, it's going to be different. Same thing happens with genes, depending on where it goes into the genome. Okay? Now, this is a graph of results from RT-qPCR, or reverse transcriptase quantitative polymerase chain reactions. And it's a way that we look at gene expression. And we're going to look at a gene, a lactase gene, that's actually in both Chinese and American chestnut, but they're expressed differently. In American chestnut, it has a low levels of expression. In Chinese chestnut, it has much higher levels of expression. Now, these are all transgenic events produced from this transgenic American here that has added this lactase gene with a constitutive promoter, so you can get higher expression. And these are all different events, and what you see is you get different levels of expression. Okay? So what you can do with this, the great thing about that is when you do a transformation experiment, you could pick low levels of expression, you can pick some from moderate levels, and you can pick some from high levels. And you can take those through your tests and find out which is the best level of expression for the outcome you want. Okay? So that's what events are and why they're useful. Okay? Now, getting back to the transformation, once we have transformed embryo cultures, we take them through several different stages that kind of mimic what happens inside of a seed. They go through all the different developmental stages. Sometimes you can't really define some of them. That's what we call the ugly stage. And finally, you get shoots. And once you get shoots, you can multiply them up and actually make a whole forest of clones from those shoots. But these shoots cannot be planted out, obviously, because they have no roots yet. They have no cuticle on the leaves. So we have to go through a rooting process where we dip them in hormones, put them in charcoal, put them back in this media, and eventually what you have is what's called a plantlet. Okay? These plantlets are pretty good, but they're still not ready to go out in the soil yet. They don't have any cuticle and no root hairs, and they would desiccate very quickly outside of high humidity. So we do a process called acclimatization. We have a growth chamber where we slowly reduce the humidity while increasing the light, and eventually get acclimatized plants, which can go in the greenhouse. Our first transgenic trees are actually planted in the field in 2006. So we've had transgenic trees since 2006 in the field. Okay, so that's getting the genes in. So what genes do we test? Well, the obvious place, what most people say, is why don't you just get the genes from Chinese chestnut, okay, or one of the Asian chestnuts? And we are looking for those. The problem with those genes is that they're quantitative. They contribute to quantitative resistance. Again, not one gene will be all you need for resistance. But we are looking at those. We're looking at 21 genes from C. millissima, 6 genes from C. seguinii, and we're also looking at 6 genes from other plants. Now, we are starting to identify some Chinese chestnut genes that look very interesting. We have a lactase-like protein, a proline-rich protein, and a lipid transfer protein genes that tend to enhance blight resistance slightly, okay? And possibly these could be the genes that are contributing together for that quantitative type of resistance. But what we're finding, though, is these genes from other plants, and specifically this one from wheat, is actually conferring very high levels of resistance. And the one that we're most interested in is the oxalate oxidase gene because it can outpace any of these other genes that we're working with as far as resistance goes. Okay. So why did we choose this gene from wheat? Well, there's actually lots of reasons. Part of it's from my background working with the pathogen. We know that Cryphonectria parasitica, and as I mentioned before, one of the things it needs to do is produce this oxalate to form cankers. And we know that because of the study of hypervirulence. In hypervirulent fungi, which are basically the same fungus only with a virus particle in it, or a hypovirus, it will reduce production of oxalate. Now, why is that important? Well, without the oxalate, you do not form cankers. This is what we call the apple test, and these are isogenic lines of the fungus inoculated on here. I just want to show you what the effect of the oxalate it has. Okay, so what you're seeing here, and it's quite obvious that if you have high oxalate, you get a lot more necrosis, low oxalate, low amount of necrosis. And actually a postdoctoral fellow, Dr. Chen, had actually done a mutation, a knockout mutation in this fungus where he prevented any oxalate from being produced and sent some of that to us, and we inoculated them on some stems, and sure enough, you actually have no cankers forming whatsoever if you can get rid of it. So the idea here is, can we design a mechanism where the plant can get rid of this oxalate? And that's why the oxalate oxidase gene is perfect for that. Okay? Now, what this gene will do is it will take oxalate and it converts it to hydrogen peroxide and carbon dioxide. Both these compounds can be utilized by a plant. Hydrogen peroxide for making lignin, carbon dioxide, of course, for making sugars, or it can just be expelled as waste. Okay? Now, the neat thing about this particular gene that we really like is that it's not a pesticide. It has no cytoactivity. It does not kill the fungus. All it's doing is protecting. So it's not considered a pesticide. It's considered a plant-incorporated protectant, or a PIP, because it is protecting the plant. Now, why is that important? It's important because it takes away most of the selective pressure for that fungus to overcome this resistance, okay? Because it can still survive on the bark of the tree and still reproduce, and therefore there's not a big selective pressure for it to select for mutations that might overcome it. In fact, it will grow on this tree just like it does on oak trees, because it can grow on the bark of oak trees also. So it's basically changing the lifestyle of this fungus from a pathogen to a saprophyte. Okay, so I think that's kind of cool. Now, another thing we like about this is just one other feature we like, is that the wheat oxidase gene is eaten by billions of people every day. Okay, we can actually go out and we get wheat germ from our store. We use that as our positive assay for this particular enzyme. So you're eating it all the time. So the question is, when's the last time you've eaten oxalate oxidase? Okay, probably right back there on the table. And the other thing, and people always ask me this, well, if you're putting a wheat gene in, what about gluten? This is not one of the genes that are involved in gluten, so it's gluten-free, okay? All right, so one of the things that the EPA asks us as we've talked to them, they always want to know, well, can you easily identify these trees? If they're going to be just like the American chestnut tree, except for blight resistant, how can you tell the difference in the forest? Well, besides one's dying and one's not. Well, we actually do have a quick assay for this, and we actually use wheat as our positive control. You can take just a leaf disc and put it in a 2-tube assay, let it sit in this assay for about 4 hours to 12 hours, and if it has the enzyme, it develops this ring. If it doesn't, it will not develop it, so you can easily identify which trees are actually expressing this gene. You can't do that with any of the other genes we've looked at so far. There's no easy assays for any of them, but this one has a very easy assay. Okay, so how do we test for blight resistance then? This is the key thing. Typically, and this is what the breeding program does, they grow trees to be about 4 years old, so they're about 1.5 inches in diameter, and then they actually take a plug of mycelia and put it in a wound on the tree, and they measure the growth of the canker over about 4 months or so. All right, so that's a very long time to wait to find out if your trees are actually resistant. So we've developed a surrogate type of method where you actually take leaves, and leaves aren't normally infected by chestnut blight, but the leaves can be used to determine if there's resistance in the tree. And the way we do that is we take the fungus, we take a nice healthy leaf, we make wounds on it of equal size, we put the fungus on those leaves, we put it in a humid chamber for about 3 to 7 days, depending on the size of the leaf, and we measure the amount of necrosis. Originally we were just measuring the length, now we're doing area, and that area is related to how resistant the tree is. Okay? So basically, this method takes about 4 years off the old method. Okay? We're actually trying to even get it even sooner. We are starting to do some tissue culture leaves to see if we can determine resistance there. And actually, the tissue culture leaves, you can tell high levels of resistance and low levels of resistance. We're still trying to optimize to see if we can find out intermediate levels. Okay. So, using a leaf assay, one of the first trees that we saw had enhanced resistance is our Darling 4 event. All right? And if you would look at the necrosis, the American chestnut has a lot of necrosis on those leaves. The Chinese has less, and the Darling 4 has intermediate levels. These are the trees that we've actually planted out for the public to see. We have some in front of our library. We actually have some of these trees out at the New York Botanical Gardens. So if you're ever out there, you can go out there and look at those trees. Okay. Now, to confirm that this leaf assay actually predicts levels of resistance, these same trees were tested under normal stem assays in the field. And what we saw back in 2012 is American chestnut, you get nice big cankers. Chinese chestnut, you get very small cankers. And the Darling 4, you also got a small canker. Okay, this is replicates of 6 trees in each type. Okay, we repeated this the following year. This year, it was slightly different results, but actually came out closer to where our leaf assays are. This year was actually a better year for growing trees, so they're much less stressed. That might be why the American has a much smaller canker on it this year. But you can see that it comes out intermediate here, just like in the leaf assay. So these leaf assays seem to be very good at predicting the levels of resistance. Okay? Now, we've made better trees than those first Darling 4s. And what's different about them is the oxalate oxidase that we are making. We changed the promoter in it, so it now expresses at much higher levels. So here's the Darling 4 level expression using RT-qPCR. It's set at 1. The Darling 215 here is about 40-fold higher. The Darling 311 is about 140-fold higher. This is our quick screen. That's why there's no error bars on it. This is just what we do when we are selecting which events to move forward. We will do better expression studies later. But the neat thing about this is that if you look at the levels of expression and you look at the levels of resistance, this is the leaf assay again, looking at it a little bit differently. The blue line represents the American chestnut, and we've normalized it to 1. The Chinese chestnuts are the orange bars, and the green bars here are the test plants. So here's Darling 4 falling between the American and the Chinese. Here's 215, actually not significantly different than the Chinese. And here's 311 that appears to be more resistant than the Chinese. And it matches with the trend of how much of this enzyme or this gene is being expressed. Okay, so that's the leaf assay. What about stem assays? Well, we haven't gotten to the full size yet in the field, but we can do what's called a small stem assay. And the small stem assay is actually a very rigorous assay in that you have to have very high levels of resistance even to survive that assay. And I'm going to show you one of them here that's been taken by time-lapse photography. Okay. This is the wild-type American, LS1. These are the two transgenic lines, which are clonal to that, except for they have the oxalate oxidase gene. And this is the Chinese chestnut control, which is resistant, susceptible, and we'll see what happens to those. This is over about 2 weeks period. They're all inoculated with the fungus on the stems. So what you're seeing is what will happen to all American chestnut trees if they're infected with chestnut blight. They will get girdled and wilt. The Chinese chestnut survives, even in this small stem assay. But look over here. Here are the transgenic ones. They are surviving just like the Chinese now. Okay? And again, these are clonal to that one there. All right? All right. So if you took those plants, and we repeated that several times, took those plants and actually looked at the stems from that particular assay, you see the American chestnut has a big canker on it, and the stem all turned black and died. The Chinese chestnut actually has quite a bit of damage on it, but the stem did survive. There's a green area on the back side of that. But here's the wounds for the two American, the Darling 215 and Darling 311 Americans, and you see there's actually moderate amounts of damage here, actually less than the Chinese. And that's why sometimes you might hear us say, well, they're as resistant and possibly more resistant than the Chinese chestnut. And that's the reason why we say that. Okay, so now we have some blight-resistant trees. What do we do next? Well, these trees, remember, they're all coming from a single cell, right? So they're all going to be clonal. They're not going to have very much genetic diversity. So one of the things we want to do is outcross our transgenic trees to wild-type trees to try to recapture the genetic diversity that's surviving out there. And we've already shown we can do this through one generation. So we've taken some of our transgenic trees. We produce pollen from them. We actually done this in a highlight growth chamber, so we've shortened the time to get to pollen to about a year or less. And we've taken that pollen, we crossed it to a wild type we call mother tree, and we have produced nuts from those trees, and we have grown those nuts and tested those for gene expression and with the leaf assays, and sure enough, the trait and the gene expression is inherited as expected. Okay. Now, another reason why I want to go to this nut production is because tissue culture plants, when you plant them out in the field, they don't do very well. Okay, they grow much slower than a seedling would. This comparison here, this is the same amount of time as this seedling. This is a transgenic seedling. This is a transgenic tissue culture plant. These grow much better, much straighter. These sometimes think they're branches and tend to grow off to the side. That can be corrected after a few years by coppicing them and then let them re-sprout. But that just adds more time to their production. So we're going to try to move from producing trees from tissue culture, which is what we're doing right now, to producing more and more trees through nuts and use those in the future. Okay, I do want to mention, you see a person back there, that we are doing environmental impact studies, a whole number of them. We've, on our second biotechnology risk assessment grant, we're looking at mycorrhizal colonization, which we're not seeing any difference. Matter of fact, we just got a paper accepted into applied environmental microbiology for that. Looking at insect feeding, allopathy, leaf decomposition, and metabolomics. And so far, everything's looking good, okay? That we're not making significant changes. So we are doing that type of work. I do want to mention, okay, where do we plant these trees once we finally get them through the regulatory process? We have to go through the USDA, EPA, and FDA to get these approved, which is probably going to be about a 5-year process. But where are we going to plant them? We don't want to cut down the oaks that are out there now. We don't have any plans to cut down trees, to plant chestnut trees. But there are a lot of places where you can plant trees. This is one example on mine reclamation sites. That's a great place. Historic sites will probably have places to put in trees. Private landowners might have old fields or something like that they want to plant chestnut trees. Also, because we're losing trees to different diseases, like in Syracuse right now we're losing all our ash trees to emerald ash borer, there's going to be opening spaces where you can put maybe chestnuts in. So there are going to be places. Now this is not going to be quick, okay? We're not going to have these things deregulated and then hand them out to people, and all of a sudden in 10 years we're going to have chestnuts everywhere. This is more like a century-long type of restoration program, and it's going to take generations. So it's not only you here, but it's going to be your children and your grandchildren. So that's the kind of restoration. It's not going to be fast, but it's going to be exciting. Okay? Now, do you think this project is important? If you do, okay, I'm going to ask you to do something. Because people always ask me afterwards, well, what can I do to help the American Chestnut Project? I want you to get out your smartphone if you have one. Okay? I did this with my class the other day, and it worked really well. I'll see how it does with this group. Now, what I'm going to do is I'm not going to ask you to donate any money or anything like that. Okay? I'm going to put that right across, even though you're going to see a button that says donate. Instead, what I'm looking for is moral support here. Okay? So get out your smartphone, and we're having this thing called a 10,000 Chestnut Challenge. And in the 10,000 Chestnut Challenge, we're trying to produce 10,000 trees over the next 5 years as we go through the regulatory process, so that when we finally get permission to hand these trees out to the public, we don't have 10 trees. We want to have a lot of trees. We want to have thousands of them so we can actually pass them out to people. So we're starting the project to try to produce those. So if you have your cell phone out or your smartphone, go to www.esf.edu, very easy, and go to this logo and click on it. And that will take you to the website. Now, when you go to the website, you're going to see this, and you're going to say, oh, donate, I want to donate. Well, that's OK. You don't have to donate right now. What I want you to do is go to that, and I want you to scroll down. If you scroll down all the way to the very, very bottom, there's a button that says Support ESF. And if you click on that button, it will allow you to put this on your Facebook page. Okay? And this will allow us to get the word out of what we're doing, all right, to everybody. Now, let me just show you the actual computer page of what's going on right now. Right now, you can do the same thing even on a computer by coming to supporter up here. Right now, we have 467 supporters. But that's kind of misleading. We actually have 1,731 people who posted this on their Facebook page. And that's done in, see, we're at 14 days left. That means we're halfway through. So this is in 2 weeks' time. We've had that many people post this for us. Okay? And according to Facebook, that translates into over 80,000 people seeing this program. Okay? So if you want to help out today, and only if you think this is important, I'd like you to post this on your Facebook. Okay? And I'm going to stop there and thank you for coming, and I will answer any questions you may have. Okay?